The isolation of functional protein families in amino acid sequence space. Evidence for design. The origin of biological complexity is a profound question that defeats naturalistic mechanistic mechanisms. One of the most striking areas of biology that underscores the intricacy of life is the study of proteins, which are the molecular machines that drive almost every process in living organisms. Proteins are made up of long chains of amino acids, and their functionality depends critically on the precise arrangement of these amino acids. Recent research into the isolation of functional protein families within the vast amino acid sequence space provides powerful evidence that life is not the product of random, unguided processes but rather the result of intelligent design. Here, we will explore the concept of amino acid sequence space, explain the isolation of functional protein families, and discuss why this points to design rather than random evolution. I will argue that the extreme rarity of functional proteins in sequence space, combined with the specified complexity required for their function, suggests the hand of a designer, God, who created life with purpose and intent. 1. Understanding amino acid sequence space. To grasp the concept of protein isolation in sequence space, we must first understand what amino acid sequence space is. Proteins are made up of chains of amino acids, of which there are 20 different types. The order, or sequence, of these amino acids determines the three-dimensional structure of the protein, which in turn determines its function. The possible combinations of amino acids in a protein are enormous. Imagine every possible arrangement of 20 amino acids strung together in a sequence of hundreds or thousands of positions. The collection of all possible sequences of amino acids is referred to as amino acid sequence space. For a protein of 100 amino acids, the number of possible sequences is 20 to the power of 100, which is an astronomically large number. Only a tiny fraction of these sequences fold into functional proteins capable of performing specific biological tasks. Most sequences either fail to fold properly or form useless, dysfunctional structures. This raises a significant problem for Darwinian evolution. If functional proteins are so rare in sequence space, how could random mutations and natural selection possibly find these functional sequences through a blind search? The extreme rarity of functional proteins suggests that they are isolated in sequence space, with vast distances of non-functional sequences separating them. This isolation presents a major challenge to the Darwinian model of gradual, step-by-step evolution. 2. The isolation of functional protein families. Protein families are groups of proteins that share a common structure and function. Despite their similarities, each protein within a family may perform slightly different tasks or exist in different organisms. However, research has shown that these families are highly isolated in amino acid sequence space. In other words, the functional sequences that define one family of proteins are far removed from the functional sequences that define another family. The pathways connecting different families are often non-functional, making it highly improbable that random mutations could bridge the gaps between these families. This isolation becomes even more pronounced when we consider specificity. A protein's function depends not only on its overall structure but also on the precise interactions between its amino acids, which must be fine-tuned for the protein to perform its specific task. This fine-tuning means that even small changes to a protein's sequence can result in a loss of function. For example, enzymes, which are proteins that catalyze chemical reactions, often require a precise arrangement of amino acids at their active site to bind to their substrates and carry out their function. If the sequence is altered even slightly, the enzyme may no longer be able to perform its task. This degree of specificity makes the evolutionary search for new protein families through random mutations incredibly unlikely. 3. The improbability of evolving functional proteins. The sheer size of amino acid sequence space and the isolation of functional proteins within it highlight the improbability of evolving functional proteins through random mutation and natural selection. To appreciate this, consider a simple analogy. Imagine trying to find a specific word in a book where every possible combination of letters is written out on every page. Most combinations would be meaningless, and only a tiny fraction would form coherent words. In the same way, the vast majority of amino acid sequences are non-functional, with only a small number forming functional proteins. Studies have shown that the likelihood of finding a functional protein by randomly searching sequence space is extremely low. The scientific literature shows that only 1 in 10 to the power of 63 sequences of 92 amino acids forms a functional protein. This number is so small that it defies comprehension, making it empirically impossible that random mutations could stumble upon these functional sequences by chance, especially within the limited time and resources available in the history of life on Earth. 4. The failure of evolutionary explanations. Darwinian evolution posits that life arose and diversified through the gradual accumulation of small, beneficial mutations preserved by natural selection. However, the isolation of functional proteins in sequence space presents a significant challenge to this view. There are several reasons why evolutionary mechanisms fail to account for the origin of functional protein families. No gradual pathways between functional proteins. For evolution to work, there must be a gradual pathway from one functional protein to another, with each intermediate step providing a survival advantage. However, the isolation of protein families in sequence space means that there are no such gradual pathways. The vast majority of intermediate sequences between one functional protein and another are non-functional. This means that evolution would have no way of traversing the gaps between different protein families, as natural selection can only preserve changes that provide an immediate functional benefit. The waiting time problem. Even if random mutations could occasionally produce a functional protein, the time required for this to happen is astronomically long. Given the extreme rarity of functional sequences in amino acid sequence space, the probability of finding a new functional protein by random mutation is so low that it exceeds the available time since the origin of life. This is known as the waiting time problem. The time required for random mutations to produce functional proteins is far longer than the time available for evolutionary processes to have created the diversity of life we see today. 5. Intelligent design as the best explanation. The isolation of functional protein families in amino acid sequence space, combined with the extreme improbability of evolving functional proteins through random mutation, points to the need for intelligent design. Proteins are highly specific, functional molecules that require precise arrangements of amino acids to carry out their tasks. The fine-tuning observed in protein sequences suggests that they were not the result of random processes, but the product of intentional design. Just as a computer program requires a programmer to write the correct code, or a watch requires a watchmaker to arrange its gears and springs into a functional timepiece, proteins require a designer to arrange their amino acids into sequences that produce functional molecules. The isolation of functional protein families is best explained by the guiding hand of a designer who crafted these molecules with purpose and foresight. From a Christian perspective, this designer is God, who created life with wisdom and intention. The Bible teaches that God is the creator of all things, and the complexity of proteins reflects his handiwork. Psalm chapter 139 verse 14 declares, "I praise you because I am fearfully and wonderfully made. Your works are wonderful, I know that full well." The isolation and specificity of functional proteins point to the creative power of God, who designed life to function in harmony with his created order. Conclusion. The isolation of functional proteins points to design. In conclusion, the isolation of functional protein families in amino acid sequence space presents a significant challenge to Darwinian evolution. The extreme rarity of functional proteins, the improbability of random mutations producing new protein families, and the absence of gradual evolutionary pathways all point to the failure of naturalistic explanations for the origin of proteins. Instead, the evidence strongly supports intelligent design as the best explanation for the origin of life's complexity. Proteins are finely tuned, functional molecules that reflect the work of a designer, God, who created life with wisdom and purpose. As we continue to explore the intricacies of the biological world, it becomes increasingly clear that life is not the product of chance but the result of divine creation. Thanks for watching. Please like, share and subscribe, and comment below if you agree or would like to add your thoughts. If you would like me to respond to your comment, please make a level 1 comment directly below the video. I will respond to all polite level 1 comments even if it takes me a while, due to other commitments.